What if understanding the brain and spinal cord requires thinking like a mechanical engineer? In this episode of Neurocareers Doing the Impossible, we introduce an emerging and a powerful field, neuromechanics, the study of how mechanical forces shape the structure, function, and pathology of the nervous system. Our guests are Silvia Budday, professor of continuum mechanics and biomechanics at Friedrich Alexander Universität Erlangen, Nuremberg, FAU, and Oskar Neumann, a doctoral candidate in neuromechanics whose academic path into neuroscience began in an unexpected place, civil engineering. Neuromechanics asks a critical question. How does nervous tissue behave when mechanical forces act on it? The brain and spinal cord are among the softest tissues in the human body. Yet, they're constantly exposed to forces, from development and growth to trauma, surgery, and disease. Understanding these forces is essential for studying spinal cord injury, brain pathology, and recovery process. Professor Budday leads the Amy Noether research group, Brainiacs, and contributes to large collaborative efforts such as CRC 1540, Exploring Brain Mechanics, EBM. In this project, mechanics is treated as an active biological signal. Mechanical forces influence development, degeneration, remyelination, and tissue response after central nervous system injury. Oskar Neumann's career path reflects the interdisciplinary nature of neuromechanics itself. He earned his bachelor's degree in civil engineering, where he studied structures, loads, and material behavior. He then moved into mechanics and numerical modeling, and now positions himself clearly as a doctoral candidate in neuromechanics. In his research, Oskar uses nanoindentation, cyclic loading, and viscoelastic testing to study spinal cord tissue. His experiments show that gray and white matter behave differently. Specifically, they differ in stiffness and respond differently over time. They are also sensitive to how fast and how often forces are applied. His work helps explain why measurements of spinal cord mechanics often vary across studies. Scale, loading speed, and tissue history matter far more than previously assumed. These insights are critical for building realistic models of spinal cord injury and recovery. Together, Professor Budday and Oskar Neumann show that neuromechanics is a foundational field for anyone interested in spinal cord injury research, brain biomechanics, neurotechnology, and surgery. This episode is for anyone curious about where neuroscience meets mechanics and how unconventional paths can lead to impactful neurocareers. Welcome, Silvia, Professor Budday, and Oskar. It's a great pleasure to have you on our podcast. Thank you very much for joining us and sharing information about this exciting new field and development. Can you please briefly introduce yourselves, and then we will dive into this exciting topic.
Thank you very much. It's really a pleasure to be here, and you introduced us already quite impressively. So definitely my background is mechanical engineering, but during my bachelor's already, I decided that biomechanics is interesting, and I want to go into that direction. And then I came to the brain, was more a coincidence than anything else. But then we started looking at the brain from the perspective of mechanical engineering, or even more so continuum mechanics, and realized that we can indeed understand things that seemed to puzzle neuroscientists or medical researchers, while it was pretty clear for us from a mechanics point of view, while all the other more biological things fascinated us because we didn't know a thing about it, but then we could learn from the neuroscientists and medical researchers. And it really became evident that there is so much still to explore and still to understand in the brain, and we can only do that by combining the expertise of mechanical engineers, biologists, neuroscientists, medical doctors. And that's how I came into the field. And since I have to say, I have not worked on too many other topics or went back to the more traditional mechanical engineering like cars, because it's so exciting. And every day we learn something new, which I think is great. So since 2023, I'm now a full professor in Erlangen, and had the pleasure to had a team of many mechanical engineers, mostly mechanical engineers or civil engineers, who are just as excited as I am about researching about the brain or spinal cord. And it's a lot of fun, and we have wonderful collaborators who only make this possible, because as a mechanical engineer alone, it would be difficult to research about the brain.
Yes, absolutely. It again confirms over and over that Neuro is an interdisciplinary team. Yes, it takes a collaboration from many, many people from different areas to come together and work on it. And you mentioned your team and people working with you. And Oskar today is one of them who does an amazing work. Oskar, can you please introduce yourself to our listeners? Thank you.
Yes, hello. I can only agree to Silvia. You did a beautiful job introducing us already. So as you said, I did my initial bachelor's degree in civil engineering, then changed to mechanics because I was more interested also in the mechanical engineering side of it. And during my master's degree, I had one class in continuum mechanics where I refitted material parameters for the aorta of pigs. And back then, the field of biomechanics seemed to me more of a niche area, and I wasn't aware of all these scientists like Silvia or other colleagues in the field doing this amazing work. And after I graduated, I was lucky enough with my application to Silvia to just fall right in the time when she got the approval for the Collaborative Research Center Exploring Brain Mechanics. And it was just an open position in the project Vivo, which is focused on in silico modeling of spinal cord regeneration, which I then took. And since then, I think I also started in 23. I've been working as a part of her team, and I can just tell it's a lot of fun. The atmosphere in the team is super nice. We have a lot of people focused on experiments or simulation or modeling. So like everybody brings in their expertise, and you can always learn every day. And this together with the nice collaborations we can get from the CRC Exploring Brain Mechanics is super fun to work with. So we have people from biophysics departments, from biomathematicians. We have molecular biologists helping understand, for example, in my project, how this regeneration works in the zebrafish or in the Xenopus lepus frog. And it's challenging, especially at the beginning, to find language which is transferable from the different disciplines. So the molecular biologist, for example, that wouldn't have the scope of what you could do with numerical modeling, for example, in mechanical engineering. And at the same time, we as mechanical engineers could not 100% understand what the molecular biologists were able to do and what kind of information were there. So it was an iterative process with a lot of steps, but within the last years, I think we managed to establish really nice language between all of us in the different disciplines. And hopefully you will see some of the results soon being published. And it's a great fun to work in this CRC.
Looking forward to seeing your new results, of course. And can you please introduce a little bit your university so that our listeners could understand the settings where you are working at and what's going on there?
Yeah, we are situated in Erlangen-Nuremberg in Germany, which is 2 hours north of Munich. Munich is a city, I think, that it is even larger or even more known, but great advantage that we have at FAU, Friedrich Alexander University Erlangen-Nuremberg, is that it's a full university where we have a medical faculty, faculty of engineering, faculty of natural sciences, and even law and so on. So even ethics, which is an important part of neuromechanics, or anything that has to do with also bringing AI into the methods, is represented at our university. And due to the fact that it's still we are not a huge city, so the distances between the different departments, the different faculties, they are not too far away from each other. So there's a lot of crosstalk. We interact. We collaborate. And I experience the atmosphere here to be very special. So we not only have FAU, but also the university hospitals. And everyone is so open to including the knowledge from the different disciplines, being open, trying to understand the language of the others, what Oskar talked about before. So this is really unique. Whenever you have a problem that you cannot solve yourself and you need some other expertise and you approach a colleague here, they are enthusiastic and willing to help. So this is very special, I would say.
Yes, it is indeed. And let's try to define this emerging field, neuromechanics. How would you define it?
I would say neuromechanics is an emerging field coming from the fact that research about the brain and spinal cord has traditionally been more from the neuroscience point of view, from the medical side, because of course, when you think about mechanical engineering or mechanics, you rather think about cars, airplanes, anything that is more a machine that we want to build. Like the literal translation of mechanical engineering, the field of studies in Germany is actually build machines or building of machines. But evidence in the recent years has shown that mechanics does play a role in the brain. And to give two examples at very different scales is that other researchers in the world, and also we could contribute quite a bit in showing that the brain during development, it develops from a flat surface to a highly folded morphology that we all know, and that is so important for higher cognitive functions. So we didn't have that characteristic structure, we wouldn't have the capacity in the brain. And also when the brain is folded incorrectly, then there are disorders like epilepsy, schizophrenia, and autism. So it's very important. And we could show that it evolves due to mechanical instabilities. So basically the outer brain surface or the cortex, the outer layer that contains all the cell nuclei, so our processing units, grows much, much faster than inner layers in the brain. So it wants to expand, but it's held back by the inner layers, which then leads to compression, like mechanical compression. And eventually it's better to release that compression by folding instead of staying in that compressed state. So this is very mechanical, but not at all obvious, I think, at first sight that mechanical forces can play such a big role during development. And even at the cell scale, and that was also shown by researchers that are part of our collaborative research center exploring brain mechanics, that cells feel and respond to the mechanical environment. And for example, axons, so the nerve fibers, grow along stiffness gradients. And stiffness gradients, so they sense their environment, and stiffness gradients control how they turn. They grow faster on different stiffnesses or slower. So really at all scales, mechanics does play a role. So there's a lot that is not understood. And Oskar was working on is working on the project on spinal cord regeneration, where there's a lot that we don't understand yet, but step by step, we see more what the role of mechanics can be and potentially is. And really by using all the different methods, so experiments, but also mechanics-based modeling and simulation, we can understand what's going on and potentially even exploit these insights for new treatment strategies or diagnosis strategies. So basically, neuromechanics is that there are forces or the properties of the tissue that influence brain in healthy development, but also disease or injury.
Thank you very much, Silvia. I'm just very curious, historically, when did it start? When did people start paying attention that mechanics actually is important to understanding and our brain development and the various injuries? And where did it happen? In which lab? How recent is this field?
It depends very much, I would say. So regarding cortical folding, there's a very old work from Le Gros-Clark. He was, I think it's 1945. I'm not sure, but there was this first, or he basically wrote down, we need the expertise of mechanical engineers to understand this more. There seems to be mechanics involved, but then it got forgotten in a way, and only more recently people looked at it in depth And regarding really how the cells sense their mechanical environment and respond to it, it started in after 2000. So it's quite recent. For brain and spinal cord, as I said, it's really ongoing. Like with this initiative, we are lucky enough to be able to be part of here in Erlangen. We can look into the different processes in the brain to explore, is mechanics important? Where is it important? Where is it not? But it's, I would say in general, it's quite recent. And of course, not only us in Erlangen are working it, but more people over the world start looking into this direction.
Yes, absolutely. It looks like it's an emerging field that's started a long time ago, but was forgotten. Yes, and now it's getting traction. And like you also noted that there are several areas of applications of neuromechanics. Maybe one of you can tell our listeners more. What are those main directions that neuromechanics is applicable and is important?
Maybe I can give one small example, which Silvia already crossed during her explanations of my project. So as Silvia pointed out, it's pretty much the start of a lot of these ideas and scientific investigations. They're combining mechanical engineering with molecular biology and biomathematics. So as Silvia also pointed out, there are some scientific works and insights on the mechanical cues during spinal cord regeneration by a process called neurotexis. The axons can sense stiffness gradients in the tissue, by which they get growing towards directions where they're supposed to be. There's a really nice paper from Kosa and Franz about this in Nature Neuroscience. So there are other findings where it could be shown that, for example, in species which are able to regenerate their spinal cord after spinal cord transection, for example, as I mentioned earlier, the zebrafish or the Xenopus levis frog, that at least in adult zebrafish, the spinal cord tissue in the adjacent proximity of the lesion core increases in stiffness during the process of regeneration. Whereas for mammals, which was shown for the mice, I think, the scar tissue, which also hinders the regeneration, obviously, is softening. So there's some idea of the mechanics to play a role there and might be usable for specific therapies. So just in 25, there were a couple of papers on using these insights into constructing and modeling hydrogel tubes with specific stiffness gradients, which then might be implantable in the cavity left once spinal cord is transected between the still healthy spinal cord stumps, which then might accelerate or enhance the regeneration, even in animals which are not able to regenerate the spinal cord, as the humans, as mammals are. So this would be one field of application, which is, like we all said, pretty much in the early stages of the scientific contributions to this. But basically, the aim of my project is, besides the mechanical characterization of spinal cord, to build models in silico where we can test specific hypotheses, and from this derive knowledge, which we can then use to model and construct hydrogels, which might be able in helping during the spinal cord regeneration process. So this would be one specific field of application there. But I'm sure, Silvia, you have a couple of more examples from the brain development or neurosurgeons.
Thank you, Oskar.
Definitely regarding brain development, we currently develop models that capture how the brain folds, so macroscopically, but also concurrently how cells migrate, how they differentiate, and so on, to be able to predict how malformations evolve, because there are malformations associated with epilepsy, and then it's treatable. So, for example, when a kid suffers from that, you can take out the affected part of the brain, of the cortex, and then they are healed. Like, usually they don't have seizures anymore. But of course, you don't want to take out too much, because then you lose brain function. But you also don't want to take out too little, because then there are still the cells that fire when they shouldn't. And the kids will still suffer. And it's very difficult to see on an MRI scan or during a surgical procedure what part is affected. And there, such models can help to understand when has a mutation taken place and how much of the brain is affected by the by the mutated cells and malformation. And even during the surgery, mechanical properties, because surgeons feel the tissue, they have to find out. It's a very, very challenging field, neurosurgery. They have to know what to take out, and the brain, when you open the skull, it just deforms and is not at all in the same position as it was when you did the MRI scan before the surgery. So all of that, there's a lot of mechanics involved, large deformations, mechanical properties. So these challenges can be addressed by integrating knowledge from mechanics and mechanical engineering and the methods.
Yes, absolutely. I can attest for that. I worked for many years on epilepsy surgery team, and we had patients with cortical dysplasia. It's the malformations that probably the ones that you meant, at least one of them. And
Exactly.
yes, and then also surgery itself, like you said, brain sinks after we open the skull and we perform a surgery. We need to adjust for that to make sure we know exactly where we're positioned, yes, and synchronize it with the MRI image that we have. So there are so many things which I never thought that mechanics can be so helpful and so useful. That is amazing, and it's definitely great help and so many valuable additional information, and for neurosurgeon and for all epilepsy treatment team. That's amazing. My next question is, what are those main characteristics in the brain that we're looking for from neuromechanics perspectives? For example, Oskar mentioned stiffness. Yes. So what are others, and why stiffness is important, and what are other characteristics that we're looking at when we're looking at the tissue from neuromechanics perspective?
So maybe I can start and then you can complement, Silvia. The first years of my PhD, I was focused on a lot of experimental investigations of the stiffness. And what I've learned is that it varies a lot within a sample, within a test procedure, and obviously across the literature. And it's also not so intuitive to understand that, like from a molecular biologist side, for example, for them there's stiffness is one scalar value of one material. But there are a lot of different kinds of the stiffnesses accessible by different kinds of mechanical measurement methods. So, for example, as you mentioned in the introduction, I was using so far spherical indentation to measure the mechanical properties of a grained white matter tissue in the spinal cord. There we have a sphere, which is typically in diameter between 10 and 250 micrometers, which is attached to a really, really tiny thin cantilever with known stiffness. And this sphere we indent into the tissue up to a predefined force level or predefined displacement level. And by measuring the bending of the cantilever, of which we know the stiffness, with mechanical models, we can derive what force is expelled by the tissue during this indentation. And with this, we get a displacement force curve, which we can then use with different models to derive the mechanical properties of this tissue based on this one testing method. So there are other testing methods, for example, double plate compression and tension, where you again get a whole different set of characteristics of the tissue. You also get the tensional resistance. Then you can also apply torsional shear to the tissue. And all these different properties, together with the biophasic characteristics of brain tissue. In the brain tissue, there are pores. The pores are filled with fluid. The fluid adds the resistance of the tissue when you mechanically load it. Create a big catalog of different mechanical properties, which you have to carefully interpret if you want to apply this to different fields. And what this can be used for is one of the examples Silvia mentioned. For example, if you want to compute the deformation of the brain tissue during a surgery when a specific method is applied on the brain, then you need to know these stiffness characteristics up on all these different ways of loading the tissue. So there are a lot of different ways of mechanically assessing the tissue. And also, depending on the experimental parameters, the temperature, if you test in a liquid or in dry, the experimental duration time, all these things play a big role. The tissue degradation, because obviously it's not so easy to test the mechanical properties of brain or spinal cord tissue in a living organism. So what we usually do for calibration of our protocols is we use ex vivo tissue, which then again suffers from tissue degradation, which we obviously try to tackle by cooling it, by hydrating it. But all these influences contribute to a big variation in the reported values for these mechanical properties in the literature. So it is quite challenging to get a physically sound set of parameters which describe the whole space of stiffnesses the brain tissue and spinal tissue can have.
Thank you, Oskar. Silvia, would you add anything?
Yeah, I think Oskar already gave quite a good overview. So it is we are dealing with brain tissue and not steel. So steel you could give one Young's modulus and that's the stiffness and that's what characterizes the mechanics of the material or the mechanical properties at least at the macroscopic scale. And brain tissue is ultra soft and it's viscoelastic, it's poroelastic. So when you load it differently, you will feel a different stiffness. And maybe I can tell one anecdote from now our collaborative research center and collaborating with the neurosurgeons and the neuropathologists. And it also will give you some insights into indentation versus compression tension loading, let's say. So in the brain, we have gray and white matter. And of course, you can see it's different tissues. So you would expect they have different mechanical properties, which is also true. It doesn't always correlate with the appearance, but of course, it more correlates, I would say, with the microstructure. But the gray matter in the cortex, in outer part of the brain, is stiffer than the white matter, which sits beneath, when you do compression tension experiments at large strains. And this is also what a neurosurgeon feels, or at least like in the conversations they would tell us. But then when you do nanoindentation, at least with indenters that have a larger indenter, several millimeters, for example, then white matter would appear stiffer than the cortex outside. So and then that agrees, and Oskar has especially looked into that, how the indenter size affects the stiffness ratio, and you can indeed show that it flips, which is interesting. But then that white matter is stiffer than the cortex, that agrees with what a neuropathologist would say when they do an autopsy and they just cut the brains into slices and go over it with their fingers. So basically doing an indentation. So that just shows that, this complex mechanical characteristics, they matter. and of course, it's not too easy to understand that. And we're still in the process of understanding.
Yes, absolutely. And I think that also explains some discrepancies between the results, across different labs. Maybe you can talk a little bit about that. And I think in some of your work that you've done, you figured out why the discrepancy actually is, and you were able to explain it.
Yeah, maybe I can start on this again. So as I pointed out before already, it is obviously depending on the setup of the experimental protocol in the lab. We also have some colleagues from different universities which, for example, grow their own mice in the lab, and then it's way more controlled, the organ harvesting and the extraction process. So depending on how controlled this is, how controlled the temperature profile during extraction, then also during the storing of the samples over the course of, let's say, one day when you perform, let's say, experiments along the craniocaudal axis of the spinal cord, and you want to figure out, are there differences between the cervical spinal cord region compared to the lumbar spinal cord region? Something might happen in the tissue, right? It's extracted from the organism. There might be tissue degradation happening. There might also be, due to the process of sample preparation, certain alterations of the tissue microstructure being enforced on the samples. So some labs, for example, if the spinal cord is small enough for small enough mammals like the mice, can use a vibratome, which is a cutting device which enables you to create really, really nice smooth surfaces, which are perfectly suitable for indentation experiments with these tiny spheres of just a couple micrometers in diameter. Whereas for bigger mammals, the amount of white matter tissue in the spinal cord is so big that if you try to cut it with a vibratome, you are not able to do it anymore. We tried it a couple times. Maybe our protocol was not right there and needs further optimization. But yeah, all these different labs, all these different research groups, they have different protocols. And under these different conditions, they do the same experiment, but yeah, they end up having different values. Then you also have this discrepancy between ex vivo tests and other modalities which are able to measure the mechanical properties in vivo, such as magnetic resonance elastography, for example, or Brion microscopy, which bring out, like I pointed out earlier, a big variation, a big catalog of different stiffness values. And then it's up on the task for the mechanical engineers to interpret these values and maybe transform one to the other. So there's another study which we recently did. We combined two experimental methods. One was the indentation process, the other one was the double plate compression tension. And then we tried to compare, are these values which we fit out of these mechanical testing scenarios, do they actually match? Because I want to know, does it make sense what I do here? Or do I just measure something which is not transferable to any other testing modality? So this is also a really nice thing within the Collaborative Research Center Exploring Brain Mechanics. We have access to all these different labs and have all these different experts in atomic force microscopy, in large strain analysis, in magnetic resonance elastography. So yeah, this is a great scenario and environment where we can actually check the same samples in different testing machines and then compare, can we transfer from one to the other? And if we cannot, what might be the reason for that? But all in all, these huge discrepancies, they're just a mix out of the different experiment protocols and the different environment conditions during which is tested and also during which the samples are stored. And another example in the spinal cord area, some of the studies, they take only the tissue. Some of them, they also have the dura matter and the pia complex around the spinal cord tissue still intact around it while they test. But it's known that this complex around the dura matter, the pia matter, is way stiffer than the tissue. So if you compare this value with the measurements we do from just the spinal cord tissue by indenting just in the gray or white matter spinal cord tissue, it's the same like comparing the stiffness of a banana, which has its shell still intact, and just the banana flesh, which is way smoother. So yeah, there are a lot of factors coming into play there when you try to mechanically assess properties of brain and spinal cord tissue.
Yes, and that it's just highly nonlinear. So and that is why it's really nice to be able to combine these testing methods and experiments with nonlinear continuum mechanics, where we can have a mathematical theory that helps us to understand these processes and kind of bring all these different results together. And with these nonlinear models, for elastic effects where fluid is squeezed out or not, you can then indeed recapture many of those results that seem at first sight contradictory.
I'm very curious about what are those mechanical properties of our neural tissue that we all already agree on. Yes, that we all are on the same side. And what are those that we're still not sure and we have those contradictory results so that we need to focus and figure it out?
So it's a really good question. I wanted to ask none, but let's say the indentation properties, we at least know some ranges. So we know it's in the area of a couple hundred Pascal, the apparent modulus you can derive from indentation of gray and white matter tissue. If the gray or the white is stiffer, highly depends on the size of the used indenter there. But this is something, and correct me if I'm wrong, Silvia, I think everybody agrees on that we are in a range of a couple hundred Pascal. From my perspective, from what I read so far and from discussions with our other scientists in the field, is that there will never be a scalar value for gray and white matter, not even for one specific indenter diameter or one specific rheometer experiments. There will also always be a field depending not only on the loading mode, but also on the temporal scale of the loading, which has to do with the viscoelastic properties of the tissue. So, for example,
whether you think about an impact, like doing traumatic brain injury, that there's a very fast loading. And when we talk about brain development, what I mentioned in the beginning, very slow loading is happening for the brain tissue itself. So there is extreme scales that are relevant for brain or neuromechanics, and of course that it is different. Yeah, but I would agree with Oskar. It's like for the different loading conditions, we do by now understand, and we do understand that the indenture size has an effect. And of course, there's always uncertainty when we deal with biological tissue. So all of that, I would say, is fine. What is need to understand even better, I would say, is going a little bit from those ex vivo testing techniques to in vivo testing. So there is the magnetic resonance elastography, where you can put vibrations on the brain in an MRI scanner and then evaluate how those waves propagate and kind of estimate mechanical properties from that. But those are, of course, at small strains and higher frequencies. And I would say that will be something we should really focus on in the future, to bring those scales together. While it's possible in an animal to test to perform indentation in vivo, I guess humans don't necessarily ethically that is more difficult, and that is a challenge, but I'm sure we will eventually also understand this, and this will be very important to go from those ex vivo techniques, where I would say we are closer to really understanding what's going on, to in vivo, because there are additional effects like blood pressure, like the brain is pulsating and all of that, which we cannot take into account with the ex vivo measurements.
Yeah, that makes total sense. The closer we are to the natural conditions, yes, of our brain, the better we understand it. And this brings me to my next question about your current main focus of research of neuromechanics. What are you focusing on, and where are you heading? What are your next steps that you are planning?
Who wants to start?
You can start if you want. I think there's more from your side.
Yeah, so I think on the one hand, it's really understanding what we just talked about, really understanding the properties of the brain in a way, including in vivo measurements, so that we can really, because our expertise is continuum mechanics, and we can develop computer models, basically digital twins of the brain, which represent the brain in a certain condition, whether it's development, whether it's during a surgical procedure, whether it's during a growing tumor. We have simulated atrophy, but for all of that, we do need the mechanical properties, and we do need to understand how to make use of in vivo measurement data to then have patient-specific brain models, like computer brain models. So that is one direction. And the other direction is, I think, also understanding how cells respond, how the different scales interact, from the more macroscopic scale to the scale of cells, where really cells have been shown to react to their mechanical environment. And that will also, I think, be important to bring the research we do more towards translation, where it can be really applied in the clinics. And there we have both the neuropathology and neurosurgery, and also neuroradiology here in Erlangen, with whom we have great collaborations, where we really see that they have ideas how to make that happen, which is amazing.
Yes. And you mentioned today AI, that you also have a team of AI specialists that contribute. And as AI is developing so rapidly, and it really becomes a part of scientific work, yes, and discovery, what is the role of AI in your research, in what you do?
Yeah, so maybe I can start a little bit, but I'm sure Oskar also has maybe some more additions. So AI, or the tools of machine learning, can be extremely beneficial for us when we develop computer models. First of all, to integrate data, to analyze data. And then the continuum mechanics-based models have the disadvantages that they are quite time-consuming. They need a lot of computer power. So if you think about real-time applications in the clinic, we will definitely need to combine our approaches with AI-based approaches. So basically hybrid physics-based and data-driven models to really exploit and make use, let's say, computational power expensive models that we usually or traditionally had. Also, I think in terms of analyzing clinical data data, imaging data, microscope images, etc., it is key to use those techniques for us. Yeah. And then at different stages during our work, we need those methods to achieve what we need to achieve.
Yeah, there was most of the points I wanted to mention as well. Maybe I can also highlight one paper of Jan Hinrichsen, one of my colleagues there, which already used their neural network enhanced inverse parameter identification for exactly this speed up, which we can use there. I think important to say is that we cannot leave all the computation only on the AI models. In the end, we have to verify that certain conditions are still valid under these found parameters. And I think, if I understand it correctly, and used it for getting better starting points for numerical simulations. So to speed up sort of the first start of the numerical simulation and then do the last bits still based on physics. So we have something interpretable in the end. Yep. And aside from this, obviously, as a doctoral researcher, there are some really nice benefits from the recent advances of AI in terms of the Copilot, which you can use for coding, which is quite handy, or a literature review assistant on AI-based formats. So this was really helpful during a lot of writing of papers for sure. But in the end, you always need the human eyes to have a look on it, check everything, and then it's good to go.
Thank you so much. And now I would like to talk more about your careers and career transition from more mechanical engineering or even civil engineering, into the neuromechanics. And one of the challenges you already mentioned, I think it was you, Silvia, you mentioned that it's not metal, that we can look how forces are applied. It's a soft tissue. It's very different. So what are other challenges that you faced when you were transitioning from classical mechanical engineering into neuromechanics?
Yeah, maybe I start this time. So yeah, as Silvia pointed out already, this is something which is extremely or can be extremely challenging and also frustrating at times, especially during the experimental investigation of the tissue. Because obviously for the first time in the life of a mechanical engineer, opposed to, let's say, a molecular biologist, you have to handle really, really soft, really sensitive specimen. And there were a lot of occasions where I broke, for example, a probe of the indenter. Each is 500 euros, which is okay, it happens. But if you're in the middle of an experiment and then you break it, then you have to start all over. Or if I remember the first human donation of a spinal cord we got for testing, I was really frustrated. I think it was during a defense party was going on in a couple other rooms. And I came to Silvia and told her about the first trials, and she was encouraging me to keep on going, and this is just normal. But I was really frustrated how hard it is to work with materials like this, which you're not used to as a mechanical engineer. But yeah, with the help of all the colleagues, especially from the Collaborative Research Center, you get to do that and you succeed. And in the end, some nice papers came out or are still coming out of this. So this was, I would say, one of the biggest challenges. And another one, obviously, even though I will never be an expert in this side, is to learn a whole new set of terminologies. So when I had conversations about the in silico model of spinal cord regeneration, there were a lot of axes, terminologies, dorsal, ventral, rostral, caudal, words which I never heard before from lectures in numerics of differential equation or operational safety mechanics. So I had to learn these, and I basically built a big dictionary for myself where I could always check what does this mean or what does this mean, or prepared for specific meetings with their regeneration experts or other people more from the bio side to have these terms memorized for this moment. So yeah, mainly handling tissue and talking to different disciplines, I would say, were the biggest challenges transitioning from mechanical engineering to biomechanics from my side.
Thank you, Oskar. Silvia, do you have something to add to that?
Yeah, I also think it's exciting in a way because you learn so much new. But I have to say, I'm of course a little bit in a different situation, or like I'm still professor at the Department of Mechanical Engineering. So my teaching, like the basic teaching focuses on basic mechanics, which is not necessarily applied to the brain. Although then I give the masters or more the further advanced courses where we can really dig into neuromechanics and the exciting emerging fields. So it's always challenging because of the things that Oskar said, but we slowly learn and adapt to. And I think it's super exciting because we can only do that because we work so closely with experts that have the background of neuroscience or medical researchers. And we bring in the expertise of mechanical engineering, and that's what we know, or should know at least. And then over the years, we could also build knowledge in more than neuro fields. Yeah, so I think what one needs is interest and excitement for the field that we don't know. And at least I always am very aware of that I'm still a mechanical engineer. So I really enjoy working together with people that have been trained in the different disciplines and to learn from them still. I would say I definitely am an expert in mechanics, and by now also in brain mechanics and spinal cord mechanics, but we will always very, very closely work with experts from the different disciplines, from the different fields, to jointly advance the field.
And I'm again curious about those mechanical engineering skills. What are the main skills, the main knowledge that you are bringing into this field of neuro, yes, from your perspective? And if somebody is listening to us and they think, oh, I really would like to contribute to this neuromechanics, what should I focus on? Because those will be the main things that I will be bringing to the table.
Yeah, I think it's really taking mechanics into account, thinking that stiffness and forces can play a role for many different processes in the brain, in the spinal cord, in the CNS. That is what I would want to give to anyone working in the field. Just have in mind, it could be something to look at, and it could explain open questions that you might have when you come from a different field, not as we coming from mechanics. And like, let's say we are even more coming from nonlinear continuum mechanics, and what we there can bring is really this understanding of the nonlinear behavior, like understanding why is the tissue responding differently when I indent than when I compress it or stretch it? Why is the stiffness flipped when I do a different loading, or if I load to larger strains? This is something we can bring to the table, like the understanding where is this coming from. And another thing that we, from a nonlinear continuum mechanics point of view, can bring and offer is understanding how mechanics interact with biochemical signaling, with cells that migrate, that divide, that how is that related to growth of the tissue, brain folding, or the sensing of cells with stiffness, to really understand this interplay between mechanical factors, biochemical cues, cell behavior. So I think this is what we need to understand for the future, and this is where we're also excited in contributing.
Thank you very much, Silvia. Oskar, do you have anything to add to that?
Yeah, well, we maybe just a small summary. The basic material modeling. And I mean, we come from a side where we are taught for 5 years how to handle mechanics, what different models there are, what different loadings there can be, all these different types. And as a small example, especially the AFM area, they're highly using, which is for a lot of cases, which they are looking at, the Hertzian model theory, which has a lot of assumptions, which are not fulfilled anymore if you have larger deformations. And in one of our last publications, we could show that the nonlinear continuum mechanics model Silvia just pointed out, they diverge from these simpler models, which more the, I would say, biophysics or biophysics community uses in terms of the goodness of fit is just better from the nonlinear continuum mechanics inspired models than from simpler models, which are more in the golden standard in the bio community. So bringing this together, all this expertise in mechanics and in the bio side, it's really, really crucial to advance this field, I would say.
Thank you so much. And next question is, many students or professionals who want to transition, let's say, from engineering, from mathematics into the field of neurotechnologies, neuroscience, they ask a question: how much of neuroscience should I know to be successful? So from your practical experience, what is that amount, the minimum amount that you needed to know to actually do this work successfully?
So when I started, my knowledge was really not that great, to be honest, of the brain, of the neural area. And I would say, if you're in a master's level and you're super interested in that field, just reach out to people, and you will find that the community is really great. The people are they're loving what they do, and they're really appreciating if students show interest in joining. And most of the really important things you will learn on the fly, or you will have to adapt and learn on the fly, because as we can probably all agree, there's just so much to know about the brain and so much still unknown about the brain and the spinal cord, that you just have to dive right in and see what knowledge you actually need for your specific field. I would assume that the biological, neurological little knowledge I gained in the last years working in this field of in silico modeling of spinal cord regeneration greatly differ from the knowledge you would get, for example, as an electronical engineer shifting towards brain-computer interfaces or spinal cord computer interfaces. So it really depends on the specific niche area of interdisciplinary science you do there. So from my perspective, general interest, enthusiasm, and approach, the will to approach people is already enough. And obviously the will to learn there to set foot in this research area.
Thank you, Oskar. Silvia, would you add anything?
So I fully agree. I would exactly say the same. So what we bring is like, if you have training in a different field, that is what you bring in. And then if you have excitement for the neuro application, you will be in contact with the people who can teach you. And it is definitely a special field in a way, also looking at it as a biomechanics person. The brain is much less understood than many other organs. So, for example, when we develop computer models, as from a biomechanics point of view, for many other organs, the processes and what we need to model is known. So our only goal or our only task is to formulate mathematical equations that can capture what is happening in reality. But in the brain, so many things are not known. So we simultaneously develop the mathematical models and still try to understand what's actually happening. And this makes it so exciting, but this makes it also, I think, just start.
Thank you. And what was the most unexpected thing you learned when you started to apply the mechanical engineering methods to study neural tissue?
Maybe I can start. So I think the most surprising and also exciting realization was that even when we use quite simple models, like quite simple mathematical models, we can explain processes or features, things that medical doctors or neuroscientists have looked at. Oh, I don't know, or like I don't understand why this is happening. This is interesting. This is a feature. I don't know where it's coming from. And then for us, it's so obvious. Like everything else that they know so much about is very puzzling to us. But then this one thing that they had not understood is so obvious for us looking at it as a mechanical engineer. So that was very interesting for me to see, and so encouraging to go into that direction and keep going, and just realizing that we can contribute. Although like I have so much for us as a mechanical engineer, you don't think at first, I would say, that you could contribute to better understanding the brain, for example, because all those medical doctors, all the neuroscientists do such a great job, and they understand so much that we don't understand. So yeah, I think that's one thing.
And especially if you're in the lab and you have the tissue and you prepare the samples and you just see how ultra soft this tissue is. I mean, it deforms under the gravitational force already so much. Initially, I was like, after the first trial experiments I did in indentation, I was not expecting to gain any insights here, if I'm quite honest, because it was the variation of the results we got out of our first test with the indenter in our lab. They were so big, and there were so many things which I could not physically interpret from the mechanical tests we conducted at the start. And yeah, I was not expecting to find insights like the publication Silvia mentioned, which is currently still under revision, where we could identify where this shift between gray and white matter tissue stiffness happens, that it appears stiffer for larger indentar sizes, and gray matter then seems to be measured stiffer for the smaller indentar sizes, or that we could actually apply two different testing modalities to this really intricate tissue and get sometimes even coinciding measurement values out of this. So I was really, like, when I started after my first year, not expecting to learn these insights from applying mechanics to the brain tissue. And it's always stays surprising. You go in the lab, you test something out, and then you can actually make sense out of it, which was from the mechanical engineering side, when you used to work with steel, wood, concrete, things like this. Really unexpected to me, and but that made the journey for me so far pretty interesting.
Yes, it's very unique journey. My mom, she is 90 years old right now, but she is a civil engineer. She's not working as such already, but I've been with her. I saw the buildings and everything that she constructed, and I've never would think that this has any relevance, to the brain tissue. And Oskar, you connected, your civil engineering background to neuromechanics, which is absolutely amazing.
Yes, although the loading scenarios in civil engineering are way better defined, I would say, than they are for processes in spinal cord regeneration. So it's different, but it's similar.
That's the challenge, or that's why we can still also develop theories that have not been there before, because no one in civil engineering or mechanical engineering was working with such a special ultra soft material.
Yes, yeah. And our podcast is called Neurocareers Doing the Impossible. And my question to both of you is, was there anything that you thought of as being impossible, but you did it anyway and it became possible? And what helped it to become possible?
Maybe I can start with this one. So as I already said earlier, my project deals with the in silico modeling of spinal cord regeneration. But as Silvia pointed out before, there's a lot of knowledge missing. We know there are some mechanics which play a role in the regeneration process, but we do not know the mechanisms by which they guide regeneration, or by which they permit it, or by which they prohibited it. So after the first 2 years, I was more focusing also, which is part of the project, on the mechanical characterization of spinal cord tissue. While I was simultaneously trying to advance this first model approach. For the brain folding, for example, there are by now, thanks to also Silvia's work, a lot of different approaches and known mechanisms which guide this folding. But for the spinal cord regeneration, there's not that much out. There are some interesting works on how individual neurons might grow and mathematical models for this. But at a certain point, I would say midway through the PhD, I was not really sure if that is even possible, especially with the knowledge we have and with the data we have. And I think we are now sort of on the way of some first model approaches, which we are currently wrapping up the paper. So hopefully this is also going to be out in a couple months. And it's a small thing, but I thought it was not really possible at times. And now we managed to get something nice out of it. And yeah, it's what I'm really happy about.
Thank you, Oskar. Silvia?
Yeah, for me, it's definitely fascinating and how much we can help by bringing mechanics into the picture. And the one thing is the example of brain folding, where it's kind of the missing puzzle piece that was not understood to really understand malformations of brain development and to be able to treat associated neurological disorders. But more recently, we also are in close contact with the neurosurgeons in Erlangen. And yeah, it is fascinating how much mechanics can help, or we do see paths happening, how to help during the surgical procedure, identify what is the tissue that needs to be extracted, like what is the pathological tissue and so on. So I could not have even imagined how many aspects there are where mechanics can help and kind of solve issues that one would have thought are impossible to solve. So of course, mechanics cannot solve everything, but it is fascinating where in fields where you would think mechanics doesn't play a role, it actually can help and does play a role here and there.
Yeah, it is amazing. And how our listeners can learn more about the research that you are doing and maybe connect with you. Maybe there are some graduate students or undergraduate students, even in the vicinity, who will learn about your work and want to learn more from you. What's the best way to do that?
Yeah, maybe I can answer this, as I'm also part of the social media group of our collaborative research center. So you can find us on LinkedIn. It's Exploring Brain Mechanics. Just type that in, and there you will always be updated by new publications, mini symposia we do on conferences, or every now and then we give small project updates on what the different projects within Exploring Brain Mechanics are doing. So this is one way to follow us. And also on the website of Exploring Brain Mechanics, which is linked there, you can find our annual report, which gives you a little more insight and description into the details of research which was done there. And specifically, there's also the website of the Institute of Continuum Mechanics. So this is focusing on Silvia's group, which I'm also part of. So there you can have the same insights of also our social activities, our recent publications, our research focuses, and also thesis proposals for open positions. So you can find us there.
Yeah, that is important. And what are you looking for in students who are applying for your open positions? What are your main criteria that you're looking for?
Yeah, so definitely excitement for the work we are doing. And many or so far, we indeed have more students and graduate students who have a background in mechanical engineering or mechanics, but also biomedical engineering could be very relevant. And since we are also more recently looking into what cells do under mechanical loading, like even that direction will become more important. So molecular biology, neuroscience.
That's good to know. Thank you so much. It was an absolutely amazing conversation, new exciting field. Of course, I wish you all the best. And before we end our podcast today, is there anything you would like to tell our listeners? Maybe I didn't ask something, but you wanted to mention, or just anything that you think would be good to mention right now.
So from my side, there's actually nothing. No, it was a pleasure talking to you, Milena. The questions were really, really interesting. Yeah.
Thank you very much, Oskar. Silvia.
Okay, so about the topic we talked about, it was wonderful talking to you, and I think if there's nothing to add, maybe more, and because I know, and I'm a lot in contact with young scientists and people considering staying in academia, for example, or going to industry. But I'm a mom of 2 kids. I'm a full professor, and I just want to tell you it's possible, and you can have kids and still see and love your kids, and still, have a career. That's maybe something I would want to tell young moms, at least, or dads that want to be involved.
Yes, absolutely. Thank you so much for mentioning that, because I will support that 100%. My son was growing up with me and going to the conferences, and I think he understood brain-computer interfaces even much better than I at that point. And he is so happy right now. He is already 25, and he remembers all our conferences and trips. And to Germany also we've been. I was giving talks. And now he programs, and he creates his own programs as well. So he does a lot of cool stuff working with AI. And I think a lot of his thinking developed during this process of growing up together with me, being a scientist and building a career. So yes, I fully support what you just said, Silvia. That's absolutely possible. And it gives our kids an advantage, I think, as well, in many ways. So it's important not to be afraid of that and just do it. So thank you very much, Silvia, Oskar. It's been a great pleasure. All possible success. And I hope that we will meet in a couple of years again on our podcast to see where you are at and what are new advances and challenges that came in this field of neuromechanics. Exciting field. Thank you so much.
Thank you.
It was a pleasure to be here. Thank you so much.
Dear Neurocareers podcast listeners, thank you for joining me and my incredible guests on this exciting journey into careers in neuroscience and neurotechnologies. I hope you've been inspired by the stories of those turning groundbreaking ideas into impactful realities. If you are looking for more guidance on advancing your neurocareer, book a consultation with me, Dr. K, your podcast host and Neurocareers coach at the Institute of Neuroapproaches, the only career service dedicated specifically to the needs of professionals in neurotech. Let's take the next step toward your neurocareer success together. In addition to consultations, the Institute of Neuroapproaches offers a variety of services designed to help you thrive in neurotech. First, professional development planning. We help you create a tailored career development plan, identify knowledge gaps, and equip you with the tools needed to achieve your goals in neuroscience and neurotechnologies. Second, resume and cover letter review. Get expert feedback on how to craft documents that are specifically designed for job opportunities in the neurotech field. Third, interview preparation. Sharpen your interview skills through mock interviews and targeted feedback, including soft skills, hard skills in neurotech, BCIs, EG, MEG, live coding, and presentation preparation. 4. Networking and job search strategy. Learn how to effectively network and uncover job opportunities while standing out in the competitive neurotech job market. 5. Negotiation and salary advice. Receive expert guidance on negotiating salaries and benefits and determining a fair salary range for your desired position. All this and more can be found at www.neuroapproaches.org. Again, www.neuroapproaches.org. So, what are you waiting for? Let's navigate the path to success in the world of neurocareers together and turn the impossible into possible. We're looking forward to working with you.
Are you noticing your car insurance rate creep up even without tickets or claims? You're not alone. That's why there's Jerry, your proactive insurance assistant. Jerry handles the legwork by comparing quotes side by side from over 50 top insurers so you can confidently hit buy. No spam calls, no hidden fees. Jerry even tracks rates and alerts you when it's best to shop. Drivers who save with Jerry could save over $1,300 a year. Don't settle for higher rates. Download the Jerry app or visit jerry.ai/libsyn today. That's jerry.ai/libsyn.